Hello student, good morning to one and all. Today the topic is microbial technology, practical aspect. The topic is monitoring of bacterial growth curve. Monitoring of bacterial growth curve. The bacterial growth curve using a spectrophotometer turbidimetric determination. It is one of the method. So actually we know the monitoring the bacterial growth curve. So we know about the bacteria petri plate. It's basically its microbes are inoculated, then it will be grown in the few days, 4 to 5 days after it will be grown. Okay. My fungus also very many times will start. Bacteria will be continuously 3 days will start the growing stage. At the same time, it's livid the plate. It automatically grown and lag phase, stationary phase, and death phase. These are the growth curve. Okay. Okay. Now we discuss about the monitoring the growth curve, the bacteria. The bacteria are interesting as they are diverse. Though tiny, these unicellular life forms make it huge contributions to many systems and cycles, from helping break down food in your intestine, to making the molecule assist in all three of the carbon, phosphorus, and the nitrogen cycles. These little bacteria can accomplish big things, and surprisingly, bacteria are model organisms for each research only. Not only because of their diversity, but also they are easily contained and they reproduce quickly. When using bacteria for research, it is important to understand the track rates of bacterial growth within a sample. Okay. The inoculant bacteria in a typical laboratory setting tend to proceed through 4 distinct growth phases: lag phase, exponential phase, stationary phase, and death phase. So the pictures show that the timeline of the bacterial growth curve, the lag phase and the log phase, and the stationary phase and death phase. So 1, 2, 3, 4. Exactly, we see that method log phase, exponential, that means lag phase, log phase, exponential growth phase. So fine, and then stationary phase, it is stopping the growth, stationary phase, and final one day is going to death phase. So once again I repeat: lag phase, log phase, stationary phase, death phase. That means log phase is nothing but a exponential. So lag phase, exponential phase, stationary phase, and death phase. So lag phase means it is a growing stage, and exponential phase is log phase. That means it's straightly, it is a peak of the up to stationary phase, like that. So these are the exponential. Stationary phase is a limit, stopping the growth. It's continue with the living stage, and finally it's going to death phase. Okay, these are the pictures clear that explanation. Okay. Now, the labs of these phases provides data that can be combined into a bacterial growth curve. During the lag phase, freshly cultured bacteria adjust to the media they have been placed in or on. As they get a feel for their surrounding and nutritional option, the cells increase enzyme production and cell size accordingly. The cells have entered the exponential phase when they begin to grow and divide at a constant phase. This phase is also called as logarithmic log phase. Logarithmic phase nothing but a log phase, because growth occurs very rapidly and the cell count can reach into the millions of and billions. As such, the number of cells is calculated using logarithmic functions. The metabolic processes also occur at the constant rate and are influenced by conditions such as pH, temperature, and the properties of the medium. The phase is called as stationary phase, is marked by the plateau in growth. Cell death and proliferation are roughly equal. Nutrients are increasingly depleted, toxins accumulate, and the cell viability decreases. The death phase occurs when these conditions cause a great rate of cell mortality than cell proliferation. The population begins to decline at a once, again exponential rate. So these are all the four types of stage which is going to what it is going on, what is happened in the fourth stage in this paragraph clearly explained in the future references. So the actually bacterial growth curves are important for calculating generation time. The generation time is the time it can take for two new cells to arise from the original cell. If starting with an inoculant with a known number of bacteria, periodic sample can be done in the calculation. The calculation equation is growth total equal to T, that means time interval, by 3.3 log by B by capital B. That means the initial population to and population after time in the population. This calculation will accurately will generate the how much times in one bacteria, one species or growth factors will be answerable. Okay, the two most common classrooms methods of determining the bacterial growth are the standard plate count. That these techniques are turbidimetry measurement, and the examples of the methods include microscopic number and a membrane filter count and a nitrogen determination, cellular weight and determination, and biological biochemical activity measurement. Okay, so another one is the bacterial growth curve. The bacterial growth curve, the four phases of growth. Number one is lag and exponential and stationary and the depth phase. These are the each and every stages will implemented, and I dictate the notes also in these pages. Okay, so you can further notes will collect the page and slides. Okay, the measurement of bacterial growth also. The how do you measure the growth factors? The direct microscopic methods, and another one is viable counting method, and third one is dilutions method, and the fourth one is turbidity method. So these are all the measurement of bacterial growth. So the direct microscopic method is the slide and grid engraved on the cover slip and place the over grid, which captures the known volume of liquid. The problem is death cells are difficult to distinguish. Small cells are difficult to see. Method not suitable for the dilute samples. So everything will be one advantages, one disadvantages are there. So the finally the OD value and cell numbers will be viable counting. Okay, these are the basic steps of measurement of growth, bacterial growth. This is a final last slides. It's going to we understand the total cells in population, life and death, and each phases. The log phase is going to this color, and exponential is going to this color, and the stationary phase is 25% is death phase stages will be disappeared, color will be changed. The finally death stage is 50-50%. Some cells are remain viable. So these are the basics and easily identified in the phases. So the bacterial growth curve is understanding and easily measurable in the future references. Thank you, thanks to watching.